ABO system. We will discuss this one. But truly speaking, there are more than 30 types of blood group system. More than 30 type of blood group system. But we are concerned about ABO system, and everyone discusses about ABO system. The reason behind it, because ABO system is more susceptible to immunological reactions. What is immunological reaction? Suppose I am donating my blood group and I am having a blood group AB and I am donating it to, suppose, a person with blood group B, just hypothetically. Whether I am eligible to donate my blood group to B, is there any reaction happens or not? But truly speaking, there are many reactions which occurs in ABO system, and that's why we discuss more about ABO system. So this is the intro. So let's begin with blood group system. How we define the blood group system, how we define the ABO system. We all know that a RBC has many antigens. So human red cell contain numerous surface structures which are known as antigens. Suppose there are surface structures, and these surface structures are known as RBC antigens, and which are recognized by immune system. These antigens are recognized by immune system. Clear to everyone? And these surface structures, or we can say there are many surface structures, but one of the surface structures are called blood group antigens. Suppose this is A, this is X, this is Y, this is Z, this is C. So one of the surface antigens are known as blood group antigens. This blood group antigen will define blood group of the individual. Clear? Two more terms are there. One is agglutinogen and second is agglutinin. So agglutinogen is antigen and agglutinin is antibody. I'm sure we all know that whenever there is an antigen, a corresponding antibody will present. So this is normally happens in the body. So now let us discuss that how these antigens are formed. So mainly two types of antigens are present. First is antigen A and second is antigen B. These are two antigens and these forms, or we can say define, blood group of the person. Get it? Now let's discuss one more main thing, which is the genes for antigens for ABO system. I'm discussing ABO system. The ABO system genes are present on chromosome 9. So this is the main thing. The genes which defines the ABO system, the genes which forms the antigen for ABO system are present on the chromosome number 9. So now let us discuss the individual antigen. First of all is formation of A antigen. So formation of A antigen. Now what happens? Expression of A antigen is dependent on the presence of H genes. please keep it in mind, expression of A antigen is dependent on the presence of H gene. Very important term, H gene. So what is this H gene? We'll discuss further. So first of all, and this A antigen is a glycoprotein. Let us define the A antigen. What is this A antigen? What is the nature of this A antigen? The nature, or we can say the chemistry of this antigen, it is a glycoprotein. First, and their synthesis starts with basic precursor cells of RBC. So this is a glycoprotein, and how this forms, let us discuss. So basic precursor cells of RBC. In the basic precursor cells of RBC, synthesis of this antigen starts. Now, this H gene, I have already told the A antigen, or we can say expression of A antigen is dependent on the presence of H gene. This H gene is present on the chromosome number, chromosome number 19. I've already told that genes which define the ABO system, or which defines the antigens for ABO system, are present on the chromosome number 9, but the gene for, or we can say the H gene, is present on the chromosome number 19. Now what happens? There is a molecule, gene for L-fucosyl, please remember it, L-fucosyl transferase. This is L-fucosyl transferase. Gene for this one is expresses, and it is this enzyme transfers fucol molecule to the oligosaccharide. Let us draw a diagram, and with the help of diagram we will discuss. This is an RBC with the nucleus, of course. This is fucose molecule, and this is oligosaccharide. Okay. So this, what is the function of this fucose transferase? The function of fucose transferase is this fucose transferase enzyme transfers this fucose molecule to this oligosaccharide. So we can say fucose plus this oligosaccharide molecule equals to H substance. So H gene give birth to H substance ultimately. You can call it H substance or H antigen. Very important thing. So this is H substance. Now, this is the basic mechanism. This, the formation of H substance or H antigen. Let me write the formation of H antigen and, or we can say H substance, happens in all individuals, irrespective of the blood group. Although we are discussing the formation of A antigen, but this is the first step, which is the formation of H substance or H antigen. This happens in all persons, whether he is having blood group A, whether he is having blood group A, B or O, whatever type of blood group he has. This first basic step, that is formation of H substance, this happens in all individuals. Now, what is the next second step? The second step is to synthesize antigen A. What happens? There is an enzyme N-acetyl galactosamine. So please consider it N-acetyl galactosamine. This is an enzyme N-galactosamine transferase. Please remember it, N-acetyl galactosamine transferase. This is an enzyme. This enzyme transfers N-acetyl glucosamine to the H substance. So what happens? H substance plus N-acetyl galactosamine is equals to A antigen. So this is the basic mechanism. So we have formed H substance first, then with the help of enzyme N-acetyl galactosamine transferase, we transfer the molecule N-acetyl galactosamine to the H substance. So together they are known as A antigen. So suppose this is an RBC, and on this RBC, this is an H substance, and upon this H substance we have a cap of N-acetyl galactosamine, NAG. So together this is known as A antigen. Okay, so this finishes the formation of A antigen. Now, the genes for this may be, we all know there are two types of genes. There can be heterozygous and there can be homozygous. So similarly, in the formation of A antigens, the genes can be heterozygous and the genes can be homozygous. So phenotype is AA first, and we can say AO. So here only functional on both chromosomes, and here functional only on one chromosome. So these are the two phenotype. But in the both cases, the genotype will be AA. So this is here wrong term, phenotype. This should be genotype. There can be two types genotypes. The genotype can be AA, the genotype can be AO, whether it is a homozygous or heterozygous. If it is homozygous, it can be AA. If it is heterozygous, then it can be AO. In both the cases, the genotype will be AA. So this here we finishes the formation of A antigen. Next is formation of antigen B. Second is formation of antigen B. How antigen forms? There is an enzyme. This here also one thing is common, that is H substance. So enzyme D the galactosyl transferase, this enzyme, this galactosyl transferase, this enzyme will transfer the molecule galactose to H substance. So we can say H substance plus galactose equals to antigen B. Understood? This is the main mechanism here also. So similarly, if homozygous, if heterozygous, altogether different story. In the homozygous, we have genotype B, and in the heterozygous, we have genotype B, O. But in both the cases, the phenotype will be B only. Understood? This is the phenotype B. Now what happens? The formation of antigen B. Third is formation of antigen AB. So in these, listen carefully, what happens that in these individuals, there is genes for both A and B anytime. So here both genes are present. So gene for antigen A is also present, gene for antigen B is also present on both maternal and paternal chromosomes. So same H substance get an acetyl galactosamine and it gets converted to A antigen. Same H substance get galactose and it get converted to B antigen. So there is H substance. First it will get an acetyl galactosamine, get converted to A antigen. Same H substance get galactose and get converted into B antigen. So here both the genes are present, so the person will have A and B, both antigen, that will be called as AB antigen. Understood? So the genotype can be AB or it can be BA. In both the cases, the phenotype will be AB. Now let's discuss the formation of blood group O. Formation of blood group O. Now what happens? The persons, or we can say individuals who have blood group O, or we can say, if I am saying I am blood group O, what it means? It means in these people there is absence of genes for both A and B antigens on both maternal and paternal chromosomes. So absence of what? Absence of genes on both maternal and paternal chromosomes. For A and B. So here the H substance remains unconverted. This is the main statement. Here the H substance remains unconverted. So we have discussed three cases. In the first case, H substance plus an acetyl galactosamine, this converted into A antigen. Second is H substance plus galactose, this is B antigen. Third is H substance, it also get NH and it is also get galactose, so we got AB antigen. Sorry, we have four cases, and the fourth case is H substance will get nothing because there are no genes for A and B. So this will remain unconverted. So we have first case, we have second case, we have third case, and we have fourth case. So according to these four cases, we have four types of antigen. We have A antigen, we have B antigen, we have AB antigen, and we do not have any antigen, that is blood group O. Now let us discuss the inheritance pattern of blood groups, that how these blood groups are inherited. So these blood groups are inherited in dominant pattern. First thing is blood group system, or we can say blood groups are inherited in dominant system, or we can say dominant pattern, not system, it's dominant pattern. Inherited in the dominant pattern. Now let us, blood grouping has three genes, or we can say three genotypes. The first one is AA, or we can say AO. There can be BB, there can be BO, there can be AB, or there can be BA, or I should write like this. It can be AB or it can be BA. So we can say there are total 6 genotypes. But what about the phenotypes? Because phenotypes are defined according to the genotypes. So in the genotypes, whether we have AA or AO, first one will get A, second one will give B, third one will give AB, and the fourth one is O, because here the fourth chance is O, or we can say here OO, that will give O only. So A antigen, B antigen, AB antigen, and O antigen. So now let's discuss the inheritance pattern. A, B, this is father, this is mother. Here is phenotype. So if phenotype is A, what will be the genotype? It can be AO, suppose. If phenotype is B, then what is the genotype? BO. Okay, so here now what will be the gametes? The gametes will be A, O, B, O. Okay. So this will now this will. So what happens? There is A, B, B, O. This is AO, and this is OO. So these are offspring genotypes. Got it? So here we can see there can be AB, there can be B, there can be A, or there can be O. So these are the phenotypes we get. Now what happens? We should say that ABO antigens are present on membrane of RBC since fetal life. So since fetal life, the antigens for the A, we can say the antigens on the RBC, like antigen can be A, it can be B, it can be AB, or there should be no antigen. So they are present since fetal life. The first appears at 6th week of intrauterine life. These are the statements which we should remember. First appear in 6th week of intrauterine life. their ABO concentration reaches up to a significant level, we can say at puberty, 15 to 70 years of life. So there should be to express any phenotype, there should be minimum concentration. So their minimum concentration reaches up to a significant level to express, to show its effect. It reaches maximum at puberty. Maximum at puberty, that is 15 to 17 years of age. One more thing I should tell you, that if we are having antigen, suppose we have A antigen, B antigen, AB antigen, we have antibodies also, and these antibodies are from IgM class.